here's a red blood cell where a single red blood cell can contain up to 300 million hemoglobin molecules. So each hemoglobin molecule is a tetramer with 2 alpha and 2 beta subunits. So you've got your 2 alpha and 2 beta subunits, and each subunit has a heme group on it, as you can see there. And the heme group magnified up is a iron protophorin 4. So the iron atom lies in the center of the protophorin, and it's bonded to the 4 pyrrole nitrogen rings. Under normal conditions, the iron is in the ferrous oxidation state, so iron 2+, so it can form 2 additional bonds, one on each side of the heme plane. So here is the heme plane with iron having a histidine residue attached to the lower plane, and this is called the 5th coordinate site of the iron molecule, while the 6th coordinate site remains free. The 6th coordinate site is on the opposite side of the plane on the top of the iron molecule. Note that in deoxyhemoglobin, which is a hemoglobin without an oxygen molecule, iron lies slightly outside of the plane of the porphyrin. When oxygen is added, right there to the iron gets slightly smaller, and the iron molecule moves into the plane of the porphyrin. So that oxygen molecule binds to iron on the 6th coordinate site. And the bound oxygen is stabilized by forming a hydrogen bond with the distal histidine molecule right there. Next, we've got when the iron moves into the plane. So right here you see the iron molecule in the center, moves into the plane of the porphyrin. The histidine residue bound in the 5th coordinate site moves with it. So this histidine molecule is actually part of an alpha helix, which alters the interface between the alpha and beta dimers of the hemoglobin molecule, which instigates quaternary structure changes. So here we can kind of see an example of quaternary structure changes right there. So when oxygen binds to the iron molecule of the heme, it leads to a structural transition from the T state, which is the deoxyhemoglobin, to the R state, which is oxyhemoglobin. The T state has a lower O2 oxygen affinity, whereas the R state has a higher oxygen affinity. This is because upon oxygenation, one alpha beta dimer shifts with respect to the other by a rotation of 15 degrees, as noted there, which allows for increased communications between the subunits, which allows for increased binding of oxygen. This is why hemoglobin's affinity for oxygen increases as more oxygen binds to the heme group. So next we need to talk about carbon dioxide and its impact with oxygen transport. So here, oops, I need to kind of move the screen over just a little bit. So here we have our lungs with a pH of 7.4, and our red blood cells travel through the bloodstream, and they're oxygenated because the lungs oxygenate them, travel through the bloodstream to the muscles, right? And the bloodstream has a pH of 7.6, and the muscles have a pH of 7.2. And then once the hemoglobin releases its oxygen to the muscles, the hemoglobin transports CO2 back on the hemoglobin itself back to the lungs. All right, so we'll kind of dive into a little bit more how this CO2 transport on the hemoglobin affects the pH of the capillaries. So when hemoglobin arrives at the body tissue or muscle, whatever, CO2 is in the tissues and it diffuses into the red blood cells. Inside a red blood cell, the carbon dioxide reacts with water to form carbonic acid, right there, carbonic acid, and in a reaction that's catalyzed by carbonic anhydrase. So carbonic acid then dissociates to form bicarbonate and a hydrogen molecule. So this hydrogen results in a drop in the pH inside of the red cell, which is why you have a lower pH in the body tissues in the muscle than there is in the lungs or in the bloodstream, because that CO2 is causing the release of a hydrogen. So when the pH drops in the tissue, this causes hemoglobin to lose its affinity for oxygen, which causes then the release of the oxygen from the hemoglobin molecule. So the drop in the pH resulting from carbon dioxide diffusing to the red blood cells from the tissues causes that release of the hydrogen molecule. The hydrogens cause a drop in the pH, and the drop in pH subsequently causes oxygen affinity for hemoglobin to decrease, which is how the oxygen is released from the hemoglobin. Then that hemoglobin transports the CO2 back to the lungs, and then that process starts all over again. So this is what's called the Bohr effect, when hydrogen ions and carbon dioxide cause oxygen to be released in the tissues. So one thing of note is that the decreasing pH causes oxygen affinity to decrease for hemoglobin. So this is why the pH of the bloodstream is higher. So since the pH goes up, oxygen affinity is not lowered, it's actually increased. So that's why oxygen stays bound to hemoglobin while it's traveling through the bloodstream, and not until it reaches the lower pH of the muscles do the oxygen disassociate from the hemoglobin due to the lowered affinity for hemoglobin. And that is all.